guys, this is investigation for part 3, looking at osmosis in living cells. You're going to do this the same day as you do your potato inquiry, and I'm not going to give you a video for the potato inquiry. You need to think through that for yourself with your group and come up with procedures that are going to answer your experimental question, which is finding out the molarity of your unknown solution as well as the molarity of the potato. And so the problem I'll introduce to you in class the day before you do the lab, and then it's going to be up to you guys to collect data in order to answer those questions during the lab inquiry day. And then also on that inquiry day, you're going to also look at solutions on a living plant cell. And so what I'm going to just show you real quick is how to make the wet mount that you need to make in order to do that part of the lab, the part 3 that is a little bit more prescribed. But then it'll be up to you guys to figure out that inquiry part all on your own. So for this part, we're going to use something called elodea. Elodea is an aquarium plant. Bought it at PetSmart. It's not hard to come by. But you're going to use this because it goes through osmosis, or it's very quick to respond to different solutions, and so that makes it ideal for this situation. You're going to use one slide to prepare 2 wet mounts side by side, and so you're going to need either one leaf that you're going to break in half or 2 smaller leaves in order to do this. And so you're going to get your slide, and of course you're going to need your microscope, which I'll get to in just a second, and then get forceps, and you use the forceps to break off a leaf and then to also break that leaf in half. And you don't need scissors, just grab and pull and your little leaf will come off. Awesome. And then you just grab it with your fingers on one end and break it in half again. So now we have 2 little chunks of our little elodea leaf. And then put one, I'll show you guys this slide here in just a second, on each side of your slide. So it looks like that, right? We've got half the leaf on each side. Then what we're going to do is we're going to use 2 different solutions and keep straight which side is which, although if you forget, you'll be able to figure it out once you look at it underneath the microscope because the cells are responding. And you have 2 different cups of fluids, or I will give these to you. You don't need to prepare these yourself. One of them is going to have just DI water in it, and the other one is going to have salt water in it. And we know what's going to happen because we talked about this in lecture. The cell that's in the salt water, the water is going to leave the cell, and so you're going to see that the membrane pull away from the cell wall. So we know what we should see. I just want to make sure that you actually can see it in a laboratory setting. So I have a little salt water. I have to hold my slide flat or else we have a giant mess. And I put salt water on one of them, and then I get my DI water on the other one, and then I need 2 cover slips. And you use your good cover slip technique and you put it down at an angle. You put 1 cover slip on each little leaf end. Okay. And then you're ready to look at it underneath the microscope. There they are. Okay, 1 on each side, so we have salt water over here and fresh water over there. And you don't really have to wait. Once you have that slide ready to go, you should be able to see differences between them pretty quick. And so, as with all microscopes, you're going to start on your low power. Get my other slide out of there. Okay. Put your slide on your stage. And then what you're going to do in your lab notebook is to draw 2 pictures. They don't need to be beautiful, but I need to be able to see them, or be able to tell what the different parts are. You're going to draw 2 pictures, one of your elodea leaf underneath with your DI water, and then one of your elodea leaf with the salt water on it. And you should be able to see some pretty clear differences between them. As far as cell organelles are concerned, you should be able to identify cell walls. You should be able to identify cell membranes. Probably the only internal organelle you're going to be able to see is going to be the chloroplast. At least that's what I could see underneath my sample. If you think you see some other stuff, call me over. I'll take a look at it and see if we can identify additional organelles as well. But it's cool to see the chloroplast because when we get to the next unit, you guys are going to be working with chloroplast and stomata, and so it's fun for you to have an idea for what those look like. Do get stamped when you have this on your microscope. That way I know everybody did this and you're not just copying pictures off each other. I want to make sure that you can prepare a wet mount and do that. Now you only need to have 1 person per group prepare the wet mount, but everybody needs to have looked at that diagram. So call me over so that you get stamped for that, because you get points for that when you turn your lab in. So thanks for watching my video. Don't neglect your potato portion. You want to have conversations with your group before you come to class, because you're just not going to have enough time to try to figure out what you're going to do and then do it during the class period, and you have to have this set up in the first hour of class. So your homework is to figure out with your group mates how you're going to work that out, and if that means you're going to go to the internet and search or whatever it is that you're going to do, that's fine, but just come into class ready to go with that potato portion. Thanks for watching my video. Have a good day. I will see you in class soon.